Good morning, students. Now we will start our second chapter, that is sexual reproduction in flowering plants. Under that, first topic is flower, a fascinating organ of angiosperms. As you have already studied in class 10 the structure of a flower, the diagram is given on page number 20, figure 2.1. The female part that is known as pistil. It consists of stigma, style and ovary. The male part is known as stamen. It consists of anther and filament. So in this chapter we will study about the sexual reproduction in flowering plants. Topic 2.2 Prefertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Floral primordium is whole structure on which these floral parts are present. Inflorescences are formed which bears the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium. Androecium is the group of stamens. Male part के group को androecium कह रहे हैं. And the gynoecium. The group of gynoecium is the female part, which consists of stigma, style, ovary. Group of pistils is known as gynoecium. Differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ and gynoecium represents the female reproductive organ. Next is stamen, microsporangium and pollen grain. In this we will study about the structure of stamen. A typical stamen has a long and slender stalk called the filament and the terminal generally bilobed structure called anther. Male part में filament है tube like and on the top anther is present. The proximal part of the filament is attached to the thalamus. Proximal means नज़दीक का part, जो नीचे का part है, that is attached to the thalamus. Thalamus वो part जहाँ से all parts are attached. All parts of the flowers are attached to the thalamus or the petals of the flower. The number and length of the stamens are variable in flowers of different species. Stamens का कितना नंबर होगा या ovaries का कितना नंबर होगा, it depends upon species to species. If you were to collect a stamen from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram will elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is they are dithecous. Anther part में दो लोब्स प्रेजेंट हैं, इसलिए इसको डाइथिकस भी कहते हैं या बाइलोब्ड भी कह रहे हैं और इनके ऊपर कवरिंग चढ़ी हुई है. थीका मीन्स अ कवरिंग. इफ यू कैन सी 2.2b डायग्राम, ईच लोब, one lobe, this side is one lobe, this side is one lobe. These are the two lobes and they are covered with the covering, so they are known as bilobed or dithecous. Anther is bilobed or dithecous. दो पार्ट्स बने हुए हैं and they are covered with the theca layer covering. Often a longitudinal groove runs lengthwise separating the theca. बीच में line of dehiscence is present, that is known as groove, जो कि पूरे anther में present है. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of 4 microsporangia located at the corners. If we say this structure, when we cut the TS means transverse section, we observe that the structure of anther is tetragonal. 4 parts हैं, 4 sides हैं और each side में एक माइक्रोस्पोरेंजिया। माइक्रोस्पोरेंजिया क्या है? एक सर्कुलर सैक लाइक स्ट्रक्चर है जिसमें के माइक्रोस्पोर्स प्रेजेंट होंगे, बाद में माइक्रोस्पोर्स बनेंगे. So microsporangia sac-like structure which are present on the all four corners of the anther. The microsporangia develop further and become pollen sacs. ये microsporangia बाद में pollen sacs बनती है और इसी के अंदर pollen grains बनते हैं. They extend longitudinally all through the length of an anther and packed with pollen grains. Now the structure of microsporangia. In a transverse section, a typical microsporangium appears nearly circular in outline. If we see from outside, it appears as circular. It is generally surrounded by 4 wall layers. Now just watch the figure 2.3b. That is the first diagram is A is the TS of microsporangium of the young anther. Transverse section of a young anther. In that, if you magnify, you can see the 4 layers. It is surrounded by the 4 layers which are shown in B, 2.3b diagram. Enlarged view of one microsporangium showing wall layers. So the outer wall is, first one is epidermis, outermost. Second is endothecium. Inner to the endothecium is middle layer, and at the last 4th layer is tapetum. So learn the names in sequence wise. The microsporangium means sac-like structure that is surrounded by 4 layers. Outer one is epidermis. First is epidermis from the outer side. Epidermis, endothecium, middle layer and tapetum. Learn the names. And if asked from the inner side the layers, then tapetum, middle layer, endothecium, epidermis. Learn these names. It is generally, the microsporangium is surrounded by 4 wall layers. The epidermis, endothecium, middle layer and the tapetum. The outer 3 layers perform the function of protection and help in dehiscence of anther to release the pollen. Dehiscence means जब वो anther open हो जाएगा और वो pollen grains बाहर निकल जाएंगे for the purpose of pollination. ठीक है. The internal wall layer is called tapetum. It nourishes the developing pollen grain. Important question. What is the function of tapetum? It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. उसमें dense cytoplasm है क्योंकि इसको nourishment provide करना है और many nucleus present होते हैं. Can you think of how tapetal layer become binucleate? Because उसमें सेल वॉल फॉर्मेशन नहीं हो रही, न्यूक्लियस डिवाइड कर रहा है बट सेल वॉल फॉर्मेशन नहीं हो रही, जिस वजह से टेपिटम के में बाइन्यूक्लिएट, हर सेल में बाइन्यूक्लिएट सेल्स हैं. मींस हर सेल में 2 न्यूक्लियस प्रेजेंट हैं क्योंकि उनमें सेल वॉल फॉर्मेशन अभी नहीं हुई. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. जो middle part है, that is the microsporangium or sporogenous tissue. Now next is microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergoes meiotic division to form microspore tetrad. जब एंथर डेवलप हो गया, एंथर डेसिस हो गया, उसने अपने आप को खोल दिया, उसमें से पोलन ग्रेन्स आएंगे. वो ईच पोलन ग्रेन जो है उसमें 4 उसमें मियोसिस होगी और ईच पोलन ग्रेन फॉर्म, ईच माइक्रोस्पोर फॉर्म्स अ टेट्राड, जो कि what would be the polarity of the cells of the tetrad? Each will be n. Tetrad because meiosis happened, so each the polarity of the cells will be, they will be tetrad. Haploid, not tetrad, they are haploid. The polarity of the tetrad will be haploid. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one of the potential pollen or microspore mother cell. The process of formation of microspore from the pollen mother cell through meiosis is called microsporogenesis. Definition is important. माइक्रोस्पोरोजेनेसिस वो प्रोसेस है जिसमें माइक्रोस्पोर मदर सेल से माइक्रोस्पोर्स बन रहे हैं बाय द प्रोसेस ऑफ meiosis. The microspores as they are formed are arranged in a cluster of 4 cells that is tetrad. जब माइक्रोस्पोर मदर सेल में मियोसिस हुई तो वो 4 सेल बन गए और वो चारों सेल पहले इकट्ठे रहते हैं क्लस्टर में. उसके बाद वो सेपरेट होके डिफरेंट माइक्रोस्पोर्स बन जा रहे हैं. As the anther mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. अभी आप देखो, पहले आपने एंथर का स्ट्रक्चर देख लिया. एंथर के अंदर माइक्रोस्पोरेंजियम है, सैक लाइक स्ट्रक्चर, जो सर्कुलर सैक नज़र आ रहे हैं, ये माइक्रोस्पोरेंजियम. इन माइक्रोस्पोरेंजियम के अंदर माइक्रोस्पोर मदर सेल हैं. माइक्रोस्पोर मदर सेल में मियोसिस होगी तो माइक्रोस्पोर्स बनेंगे. पेज नंबर 23 पे फर्स्ट ए डायग्राम में आपको माइक्रोस्पोर दिखाया है टेट्राड. मियोसिस हो गई, अब वो चारों माइक्रोस्पोर जो हैं इकट्ठे जुड़े हुए हैं. अभी उसके बाद मेच्योरिटी होगी, वो चारों अलग हो जाएंगे. तो हमने नेक्स्ट डायग्राम में दिखाया है कि वो एक ग्रीन कलर का कि वो सेपरेट हो गया. ईच वो एक टेट्राड से अब 4 पोलन ग्रेन्स बन गए. उसमें पोलन ग्रेन में क्या है, वैक्यूल है, न्यूक्लियस है, ये स्ट्रक्चर शो की है. देन उनमें फिर डिवीजन होगी, न्यूक्लियस फिर डिवाइड करेगा तो एक वेजिटेटिव सेल और एक जनरेटिव सेल फॉर्म करेगा. पोलन ग्रेन जो 2 सेल स्टेज पे आ जाएगा, अपर वन लार्ज वाले को बोलेंगे वेजिटेटिव सेल एंड द लोअर वन इज नोन एज द जनरेटिव सेल. नाउ द स्ट्रक्चर ऑफ पोलन ग्रेन. द पोलन ग्रेन रिप्रेजेंट्स द मेल गेमेटोफाइट. If you touch the opened anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed by the variety of architecture, size, shapes, color, designs seen on the pollen grains from different species. We will continue with this. Thank you.